Hello everyone and welcome to today's video. Today we're going to be going through part 3 of the cell. So that means we're going to be going through the nucleus and then we have to go over the cell cycle. As we go through the cell cycle we have to replicate the DNA. So we're going to do a brief overview of DNA replication. Hopefully it's a reminder of something you've learned previously. Then also same with mitosis and then cell cycle. So we're going to try to relate this to anatomy and physiology and see where it's important in the body. So cell cycle control is especially important in something like cancer because cancer is a result of uncontrolled cell division. So maybe we can relate a few things to that and maybe some checkpoints that aren't processed. But first we have to start looking at the nucleus and the structure of DNA and how it's stored. So let's get right into it. All right, here we go. So right here we have the structure of the nucleus. Remember, nucleus is typically at the center of the cell, and now different cells in anatomy and physiology could have one nucleus, could have multiple nucleus. So some cardiac cells actually have multiple, and they're called multinucleate. Uninucleate is one nucleate, and anucleate is a cell that has no nucleus. So cells could have no nucleus. Red blood cells actually eject their nucleus, and then they're just completely filled with hemoglobin so they can efficiently carry oxygen. Not only that, it makes them concave in shape so that they can squeeze down and get through the small capillaries. So it's very, very efficient. Even though they don't have a nucleus, they just die eventually because there's nothing to maintain them. So here, just the overall structure of the nucleus. At the center, we have the nucleolus, which is where all the DNA is packed around the membrane here. So this is there are these nuclear pores around the membrane. Now these are kind of like the pores that we have in our plasma membrane outside our cell. So these select what comes in and what goes out. So certain things that come out, we have our messenger RNA, different enzymes and so forth. Things that come in, different transcription factors, gene expression activators, and so forth. So this is a control mechanism into what comes in from the cytoplasm and what exits here. Also here, so that is again is called the nuclear envelope. Then we have the chromatin. Chromatin is DNA. DNA plus protein. It's just what that complex is. So this can come in slightly condensed, and then we can condense it down even further into things called chromosomes. So this is just the general structure of a nucleus. This is important when we're talking about cell division as we're coming up here, because this envelope needs to degrade in order to then separate the DNA into two new daughter cells. So how does this pack down now? So right here we see the general structure of DNA. It begins wrapping around itself. It's kind of like spool wrapping around a thread. We have so much DNA that it can't be all uncondensed at the same time, or it doesn't fit. I think from one cell, if we stretched out our DNA, is about the height of a normal person. It's about 3 meters tall. So what it does is it wraps around these little proteins here called histones. Right here, this little complex where it wraps around about 150 base pairs is called a nucleosome. These nucleosomes then wrap around each other, condense further and further down, keep wrapping around these histones, and then those keep wrapping around each other, and then they condense further and further until we get to that stereotypical chromosome, which is that X shape that we see in the cell. Now, remember, this side is exactly identical to that side. This chromosome complex here is a duplicate of each side. So keep that in mind when looking at this as well. Sorry, my dog is running around by my feet right now, so you might hear him. So other parts here, when DNA becomes condensed, it can no longer go through gene expression, because if our gene is hidden in this region right here, we can't get the transcription factors out there and activate it. So this is known as heterochromatin or silent DNA. In order to activate gene expression, let's say our gene is located right here, we have to separate it out. This is called euchromatin. So then transcription factors can come in here, then we can make the messenger RNA, which we're going to talk about in the next part, for the last part of the cell for translation. Well, transcription and then translation all in the same one. Today we're just going through DNA replication. So that's just a general structure of how chromosomes are packaged into the cell, or how DNA is packaged into the cells. It has to condense down in order to fit. So now we can start going through the cell cycle. So we typically draw this on this little clock scale because it's a cycle. Now, what are the different phases here? So this is the birth of a new cell. So at the end of mitosis, which that's where we're going to end the cycle. So we go through the first phase, which is G1. This is typically the longest phase for a normal cell, one that's not dividing super fast. So this is when normal growth occurs. There's a checkpoint in this phase that says, okay, we're going to replicate the DNA now. If it doesn't go through that checkpoint, it goes through an undividing phase called G0. And things like our neurons are typically in this undividing phase because they don't divide. But if they get this checkpoint here, we're going to talk about what that checkpoint is coming up later, they enter the S phase, which is when DNA is replicated, S for synthesis. So DNA is replicated here, and now we have 2 copies of each strand of DNA, because now we need to separate it then in mitosis. And now that we have 2 copies of each strand of DNA, the nucleus is very, very busy. So if we enter S phase, we want to get through the rest of cell division. Then the cell goes into G2 phase, which is where it's just preparing for mitosis. So it's duplicating the centrosomes, and eventually those are going to move to opposite sides of the cell. And then we enter mitosis, which we're going to get into later. Remember, we have the different phases of mitosis, prophase, metaphase, anaphase, and telophase, ending with cytokinesis and the splitting of 2 daughter cells, which then go through this cycle all over again. So here, one cell splits into 2. That's just the general overview, and this is all controlled by different regulatory mechanisms, which we'll talk about coming up. So the first part of that is where we need to replicate the DNA in S phase. Hopefully this is a reminder. I'm not going to go in. I know this is very, very light on the actual details. This is far more complex than what this image shows here. But this is just a little crash course in DNA replication. So the essence of DNA replication is we need to separate the DNA. It's double-stranded. We need to break the hydrogen bonds in the middle. We do that with an enzyme called DNA helicase. It can break those hydrogen bonds, and it moves down through the strand as it's breaking those hydrogen bonds. Now, just make a note that this also forms what's called a replication bubble. So if this is the DNA strand, here is where it's separated, and there's a helicase going that way and a helicase going that way. So this is all double-stranded right there, and this is where we're going through replication then. So here, then this one is going that way, and this leading strand is going that way. So so this is called replication bubble. And these intersections right here, they're called the replication forks because they look like a fork. And that's where helicase is present. Now, also important here is the directionality. So new nucleotides are added to the 3' end. So all of these go 5'. Let me change my color back here. So 5' to 3'. This is the direction of DNA replication. So this comes in 3 stages. We have to initiate it, we have to elongate it, and we have to terminate it. So the initiation, an RNA primer comes in, latches down. So now we have RNA on DNA. The whole reason that RNA primer gets laid down is because it's what brings in the key enzyme in this replication process called DNA polymerase. DNA polymerase then adds around 50 nucleotides per second. So that's an enzyme, 50 reactions per second. That's pretty fast. And this is in humans. Bacteria actually have a DNA polymerase that does it 500 times per second. But we can replicate our entire genome very, very quickly because we open up thousands of these replication bubbles for every chromosome. So then they all come together and fuse together. But anyway, DNA polymerase then continues down the DNA strand here, adding 1 nucleotide, forming complementary base pairing. Is it perfect? No, but it does a pretty great job. There are also some checkpoints that come in later and make sure it's replicated properly. So it moves down through, everything's great. One other enzyme that's not shown here is called Topoisomerase. So as helicase is moving down through, imagine pulling 2 bread ties together that are twisted together. They're going to get tighter and tighter as that wrapping happens. So Topoisomerase actually makes little nicks up here and reattaches it to relieve the upstream winding tension from helicase unwinding the genes or the DNA. So this is the leading strand. It goes, it elongates, and then it reaches the end and terminates. It's pretty simple. Now, one thing I want to mention here is the lagging strand. So I mentioned this directionality. This directionality is very important when discussing the lagging strand. So here is the 5' end, and this is the 3' end. So we're adding nucleotides to the 3' side. That's great. But we're going to reach a part right here where we just added nucleotide. So it's going to reach this part and stop. We're continuing to open up this way, and now we need to also go this direction. So what happens is that the lagging strand gets laid down in segments. They're called Okazaki fragments. Because of this directionality and the way it can only add to the 3' end here. So every Okazaki fragment, we have to bring in an RNA polymerase. It adds a bunch of, and then DNA polymerase comes in, adds the bases, and then it moves down. Another RNA primer comes in, DNA polymerase, and it goes. So Okazaki fragments, it's all laid down in fragments. But then we have to come in with another DNA polymerase and remove all these RNA nucleotides that are there from the RNA primers. So it comes in, has exonuclease activity, removes those nucleotides, and then adds the correct DNA bases in. So it's really cool how it works. And then there's a little gap that happens, and we seal it with something called DNA ligase, which glues it together. So it makes that last little bond there, which is neat. So this was like the quick and dirty of DNA replication. There's much more to it than this. Remember, another key part is that DNA undergoes what's called semi-conservative replication, meaning one of the parent strands, so if this is a parent strand here, and this is a parent strand here, after every round of replication, it's conserved. Semi-conserved, meaning half and half. So yeah, that's pretty much DNA replication in a nutshell. And then we replicated the entire genome, and we can now separate those two ends of the chromosomes in mitosis, which is what we're going to get to now. So mitosis, and hopefully this is also a nice little review for things here. So interphase here is when we have all the DNA in the middle starting to get condensed, but you can't really see the differences. So this is showing G2 of interphase. Centrosomes are still with each other, and they're not separated yet. Then the cell enters prophase. I'll write it up here because this image isn't perfect. Prophase then is where we start seeing these condensed strands of DNA. So if you're looking at this in a microscope, you see spots appearing. You don't see these centrosomes in the microscope, though, but they start growing out microtubules and pushing against each other, and they want to orient themselves at the opposite ends of the cell, so the cell becomes polar, and we know which way to separate the DNA. So as these microtubules push against each other, these migrate to opposite poles, and then we get the prometaphase where they do reach opposite poles. So the only difference between prophase and prometaphase here, so then this is prometaphase, is that the nuclear envelope has degraded, so we have to break that down to separate the DNA, and the centrosomes are at opposite sides. Remember, each centrosome has two centrioles in it, and they're microtubule producing centers. And then we get to metaphase. Metaphase is when everything's lined up in the middle, so M for middle. So chromosomes are all lined up in the middle, and here microtubules are slowly pushing each chromosome to the middle and attaching at each end. Now, there's a checkpoint right here. This is called the M phase checkpoint. It doesn't go into anaphase until every chromosome. So if I were to draw a chromosome down here, so here is one, and here's the other side. At the middle, we have the centromere, and these microtubules attached to it, and that attachment complex is called a kinetochore. Now, we don't enter anaphase, which is the next step here, where they're pulled away until each side has a microtubule attached. That way, when we enter anaphase, then one's pulled that way and one's pulled that way. If one side isn't attached, then both chromosomes will pull one direction, and then this side won't have any. This is called non-disjunction. So this is a very, very important step in this process, very important checkpoint. So something like trisomy 21 or Down syndrome is caused by a non-disjunction, meaning one of the spindle fibers broke down and two chromosomes were brought one way and not enough chromosomes were brought the other way. So that's anaphase. They move away from each other. And also, one thing I like to note here is that these microtubules or spindle fibers, two of them are pushing against each other. what that does is that elongates the cell. Imagine pushing your hands against each other, your elbows push out. So that's kind of what this does here. So here at metaphase, we're about that wide, and anaphase, we get a little wider from that pushing out, and that pushing out helps then the next step here where we go through telophase and cytokinesis. So here, telophase is when this actin ring starts constricting around the middle of the cell. Imagine, again, I like to use bread ties as my examples. Twisting a bread tie, that's like the actin ring, and eventually the two cells will just pinch off away from each other. That DNA can then uncondense, and the cell can then enter G1. So both cells will then enter G1 and grow and undergo gene expression and do their job. So mitosis. Now, how do we control this? There are different checkpoints. So I mentioned some. We have the M checkpoint. That's where it makes sure all the spindle fibers are attached to the kinetochores, the centromeres. Here, this is the G1S checkpoint. Imagine these as like stop and go lights. You know, you have your red light, green light, and you have your red light. If it gets the green light, it can go. If it doesn't, it sits there. And this point, if it doesn't get the green light, it goes into that G0 or undividing state. And as we go around, there's also a G2M checkpoint that says, okay, we're ready to enter mitosis. So now what controls these checkpoints? These two right here. What's the main control mechanisms? And that can be described here. There's this protein called cyclin. Well, there are different subunits of the cyclin. Cyclin D, cyclin E, cyclin A, and cyclin B. Down here on the x-axis, we see the different phases. And over here on the y-axis, we see different concentrations. So do you see a unique pattern with each of these? They're cyclic, meaning they rise and fall depending on which stage they're in. And the cell actually is able to figure this out. There's another enzyme in this process called cyclin-dependent kinase, or CDK. What happens is the cyclin binds to CDK, forming the cyclin CDK complex, and that complex then activates whatever phase of the cell cycle it is for that particular cyclin. If we just look at the general overview here of cyclin D, as this concentration increases, it then binds to cyclin-dependent kinase, activating the different stages of the cell cycle until we get to the end of mitosis. So here is where we regulate those checkpoints. So entering S phase, that checkpoint, that main checkpoint is right here. So it's when these two concentrations are that high. And then we have the G2M checkpoint, and that's what it's looking for right there, those two concentrations. And then afterwards, cyclin falls off of that cyclin CDK complex, and then it gets degraded. So down here, we see all the degrading of the, well, D, A, and B, at least. E gets degraded right here. So it gets degraded, and then eventually they start building back up, and then we go through the process all over again. So it's very cyclic. And if something would go wrong in this, imagine if cyclin never came off CDK. If cyclin never came off CDK, the cell would constantly be going through cell division. And there have experiments have been done where they take the scientist takes the cytoplasm out of a cell that's in G2 phase, puts it into a cell that's in G1, and that cell automatically jumps to G2 and never replicates the DNA. So that means these protein complexes, protein enzyme complexes, are what are regulating entering mitosis then. So it's really, really neat how that works. That's not the only thing that controls cell division in these checkpoints. Here, I have another one, growth factors. For growth factors, the main one is one called PDGF, or platelet-derived growth factor. So if we had a culture of, let's say, liver cells, we gave one this growth factor, and another culture we didn't give the growth factor, the one that's given that growth factor will grow more. So cells release growth factors for repair mechanisms and so forth, and that's the platelet derived. So they're from our platelets found in our blood. So that's an important one there. And some cancers can make their own growth factors and promote growth via that mechanism. And then here, density. This is a big one for cancer. So cells want to adhere to the surroundings, but they don't want to have so many cells around them that they can't get nutrients anymore. And one thing that fails in this control mechanism is that in cancer and tumor growth, cells are dividing uncontrollably. So they're forming tons and tons of cells around them. The center of that tumor isn't getting the proper nutrients, not the proper vasculature, not the proper oxygen. So it's typically necrotic in the center of a tumor because of this density-dependent inhibition failed. So usually cells reach a certain density and they stop growing and dividing. So these signals shut down. The cell has this negative feedback mechanism and says, okay, let's not divide anymore because we don't want to starve ourselves. We don't want to suffocate ourselves. So these are the main ones that control these checkpoints. But the big one here is that cyclin CDK complex. And a lot of things in our body are determined by this platelet-derived growth factor. So that was a little quick and dirty of the cell cycle, mitosis, DNA replication, and then also the breakdown of the nucleus. Hopefully you guys got a little bit of good information today. If you have any questions, please let me know, and I'll see you next time. Thanks. Hello again. Thank you for watching my video. I'm glad you could come by. If you enjoyed this content, feel free to do that thing where you like and subscribe. Also, if you're interested, I prepare and record all of these videos live on Twitch. After the recordings, we do a post-recording question and answer section, and then sometimes I play games where any question is fair game as I game. Also, on Twitch, I'm a co-founder of a wonderful community called the Knowledge Fellowship, or TKF. Our goal is to seek and share knowledge with others while live streaming. We feature streamers who cover biology, law, physics, chemistry, geology, photography, health and fitness, psychology and mental health, rocket science and aeronautics, programming, and tons of different practical EDU streams. Feel free to check out TKF at the link in the description and join the very active Discord community. Hopefully, you find something that interests you. With that, I'll see you all next time and have a wonderful day. Remember to seek knowledge and challenge everything.